Hello everyone, this is Rubina Ahamed. I'm an MSc student in Department of Pharmaceutical Science. Welcome to my presentation on gene cloning and protein expression. Today I will discuss about the protein human carbonic anhydrase. First, I want to give you a brief outline of my presentation that I will discuss here. I will first discuss about the background of carbonic anhydrase as a drug and drug target. Then subsequently I will discuss about cloning and expression steps like ligation, transformation, bacterial cell culture, plasmid purification, verification and characterization. Let's start with the background information of carbonic anhydrase. So what basically carbonic anhydrase is? It is gene-containing metalloenzyme which contains gene in its binding site. It is also known as carbonate dehydratase. Carbonic anhydrase plays an important role in the formation of carbonic acid from carbon dioxide and water. This carbonic acid dissociates to release proton, which is very essential to create acidic environment in our body. Now I want to tell you why did I choose this protein. Because I found that this protein can be used as a drug or drug target. When it is considered to be a drug, it plays an important role in the regulation of pH and fluid balance. It also helps to make pancreatic juice alkaline and saliva neutral. Not only that, it also maintains water content in different vital organs in our body like eye, kidney, stomach. So any kind of deficiency of carbonic anhydrase can lead to disease. Therefore, replacement of carbonic anhydrase is being used as a drug. It can also be used as a drug target when carbonic anhydrase is a causative factor of disease pathophysiology. For example, glaucoma. Glaucoma, which is caused because of excessive level of carbonic anhydrase, and therefore an inhibitor of carbonic anhydrase will help to cure the disease. So considering the potentiality of this protein, I wanted to do further research on it, and I built up an experimental design to get a purified protein so that I can do my further work on it. For that one, what did I need? First, I needed to know the sequence and design messenger RNA for this protein, as messenger RNA contain the genetic information for amino acid sequence of protein. I got this messenger RNA sequence from the website NCBI. My next step was to perform PCR amplification. Usually, conventional PCR amplification is performed in two separate steps. First, I performed reverse transcription of messenger RNA species to get complementary DNA through the enzymatic activity of a reverse transcriptase in the presence of four types of DNTP and oligo-DT primer. Secondly, I performed real-time PCR amplification of complementary DNA product using gene-specific forward and reverse primer, and I designed it from Jembos. I also use heat-stable Taq polymerase, suitable buffer, and magnesium chloride. During the PCR steps, I needed to maintain a specific temperature for each of these steps. For example, I can say for the denaturation, I needed to maintain the temperature at 92 to 95 degrees Celsius, and for hybridization of polymer, the required temperature was 35 to 70 degree. And the activation of Taq polymerase, I maintained the temperature at 72 degrees Celsius. From these steps, I got my DNA sequence of gene of interest. Then what I did ligation. But before ligating the DNA to vector, I needed to purify my PCR product because sometimes non-specific amplification products are observed. For this purpose, I used agarose gel electrophoresis to get the desired length of my DNA of interest. Then I recovered it from the agarose gel. I cleaned it with PCR cleanup kit. For insertion of my DNA, I need a vector. For inserting, I used a topo-TA cloning vector here. The reason behind that, it is a very highly efficient 5 minutes one step insertion of Taq polymerase amplified PCR product into the plasmid vector. Ligation of vector with PCR product containing 3'A overhangs is very efficient and occurs spontaneously within 5 minutes at room temperature. Here you can see the diagrams that shows the concept behind the TA cloning method. Here is the map of my topo TA cloning vector, and the arrow indicates the start of transcription of T7 RNA polymerase. For a successful cloning, a vector should have some specific feature like promoter, kanamycin resistant gene, ampicillin resistant gene, T7 promoter, and priming site. I have all of these features in my topo-TA vector. For transformation, then I needed to choose a cell line. The cell line I used here was E. coli strain because it was tailored to express foreign gene. It contains a bacteriophage T7 RNA polymerase in its chromosome, and the expression of the protein was controlled by the bacteriophage T7 RNA polymerase promoter element. After ligating the insert into the vector, I used a heat shock method for the transformation of my plasmid into the E. coli cell. For that one, I mixed all the components together, cooled on a ice for 1 hour, which allow vector sticks to the cell. Then I suddenly increased the temperature to 42 degrees Celsius, which allows the vector to enter the cells via membrane. Then I again to cool them to grow up in LB plate. So transformation was done, and after transformation I allowed the bacteria to grow in a LB plate overnight. Then I got two types of colonies, white and blue. I picked white colonies for the subculture because it is most likely the result of cells transformed with a recombinant plasmid in which the vector has the PCR DNA insert. After overnight incubation, I extracted the plasmid DNA from each of these bacterial culture and analyzed it by restriction enzyme digestion. When I got my desired plasmid, I transformed it into the cell for protein expression. After overnight growth of this bacteria, I ran induction tests to see whether my protein was expressed. And I found that my protein was expressed, so that was great. After having my protein expressed, in bacterial cell, I developed a purification protocol for purification. I centrifuged the cell culture in 2 steps at different RPM to get a supernatant which contained my desired protein. Then I purified the protein by using appropriate affinity chromatography like His-Tech nickel NTA column, as my protein contained this tag. Restriction enzyme was used to clip to tag from this tag protein. After all this process, I performed SDS phase gel to determine the purity of this protein, actually for further purification. Then I performed mass spectroscopy for sequence the identity with appropriate wavelength. With all of this process, I got my desired purified protein. So now I am ready to do my further research, and my further research plan is to develop a inhibitor of carbonic anhydrase. So that is all about my today's presentation. Thank you for joining. Please let me know if you have any question. I would be so happy to provide any kind of information regarding my today's presentation. Have a good day.